What if I told you that the reason cancer keeps coming back isn't because we're not killing enough cancer cells, it's because we're killing the wrong ones. There's a small population of cells hiding in every tumor that are basically the cancer equivalent of cockroaches after a nuclear apocalypse. They survive everything we throw at them, and then they rebuild the entire tumor from scratch, but scientists just figured out how to hunt them down with radioactive bullets. Let's talk about one of cancer's most frustrating tricks. You go through months of brutal chemotherapy. The tumor shrinks, the doctors say it worked, you celebrate. And then, months or years later, the cancer comes roaring back, often more aggressive than before. This happens to thousands of people every year, and for the longest time doctors thought it was just bad luck. Every tumor contains cancer stem cells, the master architects, while regular cancer cells are like foot soldiers that can be killed relatively easily. These stem cells are the generals. They're incredibly hard to kill, they can hide for years, and they can rebuild an entire army from scratch. Cancer stem cells make up only 1 to 5% of a tumor, but they're responsible for up to 90% of cancer deaths. To understand why this is so important, imagine cancer as a dandelion. Most people try to get rid of dandelions by cutting off the visible part, but if you don't get the root system, that dandelion is coming back. Cancer stem cells are like that root system, hidden, tough, and the source of everything growing above. Cancer stem cells have superpowers. They barely divide, have efficient DNA repair systems, hide in protective niches, and can transform into any cancer cell type needed. So how do you hunt something virtually invisible and expert at hiding? Swiss researchers decided to use precision targeting and radioactive ammunition. First, they needed to identify cancer stem cells. They discovered stem cells have unique protein combinations on their surface, like ID badges only stem cells carry. They take an antibody, a bloodhound that can track specific scents, train it to recognize stem cell proteins, then attach radioactive payload. The antibody travels through your bloodstream until it finds the target cell and binds to it. Most treatments use lutetium-177, like using a sledgehammer. These researchers tested terbium-161, more like a precision scalpel. Lutetium emits beta particles like rifle bullets that travel far. Terbium emits electrons like shotgun pellets that dump energy in a tiny area with minimal collateral damage. When researchers tested both approaches, terbium-161 was significantly more toxic to cancer stem cells. But the real test came in living mice. The results were spectacular. The terbium-161 treatment eliminated all cancer stem cells. Every single one was destroyed. Even more impressive, it completely prevented tumor growth. Treated mice remained cancer-free while control groups developed tumors as expected. This could revolutionize ovarian cancer treatment. Instead of hoping for remission, we could eliminate the root cause and prevent recurrence entirely. But applications could go beyond ovarian cancer. Cancer stem cells exist in virtually every cancer type. This approach could be adapted for all of them. This could lead to truly personalized cancer treatment, custom radioimmunotherapy tailored to each patient's specific cancer stem cells. Scientists have figured out how to hunt down cancer's most dangerous cells using radioactive bullets guided by molecular bloodhounds. It's not just about treating cancer anymore, it's about preventing it from ever coming back. This research is still preclinical, but for millions facing cancer diagnoses, this represents genuine hope for truly curative treatments. What do you think about this precision targeting approach? Share your thoughts in the comments. If cutting-edge cancer research fascinates you, hit subscribe and the notification bell. Science is moving fast, and together we can stay on top of discoveries that matter most. Thanks for joining me.